Rayhan Chenoy. Rayhan is a rising senior at Del Norte High School. I'm going to tell a personal story to introduce Rayhan. So I was in the interview with his mentor, Matt Jacobs, and after Rayhan left, Matt said, "Wow, he is cool as a cucumber." And I can confirm, after 8 weeks of getting to know Rayhan, he is indeed cool as a cucumber. So please welcome to the podium, Rayhan Chenoy.
Thank you, Madison, for that introduction. I'll do my best to uphold the reputation that precedes me. Hi, everyone. My name is Rayhan Chenoy, and I work in the Systems Neurobiology Laboratory under the direction of Dr. Edward Callaway and the mentorship of Matthew Jacobs. And the topic of my research this summer was the local and long-distance circuits in the mouse visual cortex. Now, the Systems Neurobiology Laboratory primarily deals with understanding the various connections between neurons in the brain and the various pathways that neuronal signals can take. The problem that we're investigating is that the brain is a highly convoluted piece of machinery. It's more like a tangled bowl of spaghetti than a neat matrix of city streets. Now, we can take a human brain and compare it to a radio, a mess of parts, wires, transistors, and capacitors. And to understand a radio, electrical engineers reverse engineer the radio to pieces to understand the functional components and how they interact with each other. In the same way that engineers use wiring diagrams to understand a radio, the Systems Neurobiology Laboratory maps the connections of different neurons and the cortical pathways that these signals follow. This is important because a lot of disorders like epilepsy, Alzheimer's, have malfunctioning circuits, and if we can establish a baseline of how the connections are supposed to work, we can better understand what goes wrong in these diseases. Now, the Systems Neurobiology Laboratory primarily looks at the mouse visual cortex. And this is because the brain of a mouse is structurally and functionally similar when it comes to the visual cortex. And in this image, you can see the visual pathway, starting in the eye, going to the LGN or thalamus, and then to the visual cortex, the primary visual cortex, as noted as V1. And the method we use to build these wiring diagrams is also what the Systems Neurobiology Laboratory is primarily known for, the rabies viral tracing. Now, rabies viral tracing is an approach that allows a modified rabies virus to hop from one neuron to only the cells directly connected with that neuron. This is called monosynaptic tracing. By detecting where the virus ends up, we can figure out all the connections to the starter cells. This is important because if we didn't have monosynaptic tracing, then we couldn't effectively figure out which cells are connected to which starter cells. The virus moves in a retrograde fashion, and this means it moves in an opposite manner to which a neuron fires in your brain. And this is just how rabies naturally works. We use helper viruses and fluorescent tags, as I noted in this image, to highlight both the starter cells and the cells directly connected with those cells. So when it comes time for image processing, it's very easy to see the cells. And as you can see in this image, the red cells are all the monosynaptic connections, and the green cells are all the starter cells. And we can zoom in to get a closer look. This is an image of visual cortex, and the three cells that look green are not actually all green. The two outermost cells are the starter cells, the ones infected with both rabies and the helper viruses. The one in the middle was only infected with the helper virus. All the red cells that you're seeing are the cells that are monosynaptically connected to both those two starter cells. To fully understand how the connections in the visual cortex work, we have to look at how the visual cortex is structurally laid out. What you're looking at is an area map of the mouse visual cortex. You can see in the middle, we have V1, the primary visual cortex, where all visual input is filtered into. Now, surrounding V1, you have the secondary visual cortex, which are made up of regions such as LM, RL, and so on. And these different regions encode for different forms of higher visual processing. Now, we know that neurons from V1 talk to neurons from V2 when higher processing is required, but we didn't know exactly how these visual areas were laid out. So two neuroscientists, Wang and Burkhalter, set up this experiment where they performed horizontal injections of red, green, and yellow chemical dyes, as shown in the picture. After some amount of time elapsed, we can see that the same pattern was conserved throughout the visual cortex. And because this pattern was conserved, we know that since the neurons in V1 talk to neurons in V2, we can make boundaries showing each specific higher visual area. This technique was used to create an area map of the visual cortex that we still use today. So we know that neurons in the primary visual cortex talk to neurons in the secondary visual cortex, but do we know how? So there are two extreme models of how these neurons talk to each other. The first one is solitary projection, or the theory that says neurons talk to individual regions in the secondary visual cortex exclusively. The second model is manifold projection, or the idea that a neuron is like an octopus and spreads its tentacles to various regions throughout the secondary visual cortex. From these two theories, the main question arose, which theory is correct? Do V1 projecting neurons project to a single area, or do they project to many? From this question that we're investigating, there are two main methods. The first method is using viral injections and microtome sectioning. And the first step in this process is the viral injection, such as rabies viral tracing, like I already talked about. We can use other viruses as well. After we perform the viral tracing, there's a brief incubation time, and then we perfuse the brain or extract it from the mouse skull. After this, we use a microtome, which is essentially an expensive deli slicer, to section the tissue into very thin slices. And these thin slices we can then stain and then mount onto microscope slides. And using an epifluorescent microscope, we can scan these slides, and then using image processing and interns to analyze tissue and count the highlighted cells. Now, the second method is similar in a few places, and the similarities will be highlighted by green. So you can see in the second method of intrinsic signaling imaging, or ISI, and light sheet microscopy, the similarities are the viral injection and the image processing and counting. However, the differences is the ISI and the light sheet. And the advantage of this method is that we don't have to actually manually section using a microtome, but the light sheet microscopy uses optical sectioning and then reconstructs it into 3D. So I'm going to explain a little bit about the intrinsic signal imaging and the light sheet microscopy more in depth. But first I want to tell you why we use ISI in the first place. It comes down to the fact that there are many differences in the areas in the visual cortex between individual mice. So in these images, you're looking at the cortex of 4 different mice, and you can see that they all have different structural layout. The higher visual areas differ in size, shape, and position. And this is primarily why we need ISI, because a brain atlas that is consistent will not work because there are so many differences in this layout between each individual mouse. So what is ISI? You can think of ISI as an MRI, or it's very similar to a functional MRI. But instead of using protons and aligning cells in the body, ISI measures the concentration of hemoglobin and essentially makes blood flow maps. The way it works is that we attach a head frame to the top of a mouse's skull and take special images of the mouse's brain from the top as the stimulus is flashed across its eyes. And ISI essentially makes blood flow maps depending on the response of these individual regions to the stimulus. The stimulus is a low-resolution light on a monitor that can either be flashed horizontally or vertically. And different regions respond differently to the stimulus. As you can see, in these maps, the red means a higher concentration of hemoglobin, while the blue means a lower concentration of hemoglobin. Using these maps, there is a segmentation algorithm that translates the data from this information into relevant data to our investigation. And you can see those 3 maps. The horizontal retinotopy, the vertical retinotopy. Horizontal is when the stimulus is being flashed horizontally, vertical. And the anatomy picture that you see are all the cortical regions that we're looking at with the boundaries already made. So there are 2 things that I want you to take away from this. Intrinsic signaling imaging creates the boundaries of the region that we need using measuring concentration of hemoglobin. And intrinsic signaling imaging is necessary because all mice show differences in the layout of those higher visual areas. So after ISI and injections, we perfuse the brain from the mouse's skull and analyze it using light sheet microscopy. So in light sheet microscopy, no actual manual sectioning is needed. You can think of it as a CAT scan. Our end goal is to perform a 3D reconstruction of the brain tissue. And in fact, we actually put the whole sample of brain tissue in the light sheet microscope. So the light sheet microscope optically sections a tissue perpendicular to the viewer, and then we can take these sections and using software, we can reconstruct it into a 3D model of the visual cortex. So we use light sheet microscope because it's unparalleled in its reconstruction quality. There is an alternative of using the epifluorescent microscope, but the reconstruction can be inaccurate because the optical sectioning that the light sheet microscope uses is just unmatched. And now I'll show you an example of an actual reconstruction using light sheet microscopy. So this is a sample of visual cortex, and you can see all this green is blood vessels, and the red are the actual cells that we're looking at. Here's another example, same thing, just a bigger sample of tissue. After both the intrinsic signaling imaging area maps are created and the 3D reconstruction of the tissue is finished, we can then work to align the images so the next phase of data collection can commence. So now that I've talked about both methods of investigation, you may be wondering why we use one over the other, and it comes down to the pros and cons. So microtome sectioning, it's very straightforward, and it's common. It's a tried and true method, and it's choice for most of the experiments so far. The disadvantages, however, is that it's extremely tedious. It takes a long time to actually section the visual cortex into thin slices with the microtome, and an annoying amount of time to mount those tissue delicately onto microscope slides. And of course, the microscope takes hours to scan. Sectioning and mounting requires a certain amount of finesse, and it's pretty easy to damage the tissue, as I discovered many times. The tissue is 1/3 as thin as a piece of paper, and it's easily poked with fibers of a paintbrush in mounting. And of course, the damage of tissue can easily compromise the experiment. The advantages of using the second method of ISI and light sheet is that the optical sectioning and the use of software is highly accurate. Also, since a lot of this method uses a computer, we can minimize a lot of human error involved in this process. It's the most advanced solution we have, and it's very promising for future experiments. However, blood vessel alignment is exceedingly difficult. It's quite painful, it has to be done by eye, and the blood vessels are very amorphous. And this step has been a serious setback for us in using the light sheet method. As of now, there's no getting around this step. Also, light sheet is very cumbersome. At this moment in time, there's a need for more computer processing power to make it a more effective method. So the final step of this process that both methods have in common is the data collection. And I'll give you a little demonstration of how the cell counting works. All the blue that you're seeing is cortical tissue. And specifically, this is the visual cortex. So we can make layers of the tissue, shown by green with a drawing tool. And you can just eyeball it. You can see that the top is a little darker than the bottom. Or for more inexperienced neuroscientists like myself, we use brain atlases, so we can have like a transparent image and actually make those boundary lines. And in this example, the red cells are what we're counting. In this case, it's pretty easy to see the red cells, but sometimes they can be very hazy, and it just can't be done using a simple algorithm. We have to count them. So this is our data, and it's preliminary, but it does address the fundamental question. And the answer is that the majority of V1 projection neurons use that theory of solitary projection, where they project just to a single area. So these graphs are of 3 different brains using the first method of microtome sectioning. And the bars show the number of V1 neurons that connect to each region. So you can see the secondary, the higher visual area in the x-axis. And the y-axis is the percent. So the number of neurons that connect to a specific region over the total amount of neurons. And bars that are labeled with 2 or more regions show co-localization. So if a V1 neuron connects to both LM and PM, it will show that in the graph. So because this column is low compared to the other columns of individual regions, we can conclude that V1 projecting neurons are exclusive, for the majority of time, are exclusive to single V2 regions. Now, the next steps that arise from this investigation is first, just to collect more data through the light sheet microscopy method. As you can see in the last slide, all the data that we have so far is using the first method of manual sectioning using a microtome. The next step after that would be to ask a new set of questions about the functional roles of the V2 regions. For example, what they encode for. So if I see a ping pong ball moving and I recognize that I recognize the motion, a specific V2 region will encode for that. Same as I can recognize this object as a laser pointer. There is a V2 region that encodes for object recognition. So once we have an idea of the connectivity in the visual cortex, we can look at these kind of questions. And all of this goes into understanding the wiring diagrams, basically understanding how all these functional components connect with each other and the different cortical circuitry. And especially, big picture, in order to identify the malfunctioning circuits in disorders like Alzheimer's, epilepsy, and depression. And the ultimate goal, of course, is something far greater than curing neurodegenerative disease. It's for my mentor, Matt Jacobs, to win the Nobel Prize. And yes, you're reading that correctly. It's 2018. I can say I have some amount of clairvoyance. All jokes aside, there are a lot of people I want to thank. I promise it won't be an Academy Award speech. But Dr. Callaway, thank you for having me. It was a great experience working in such a high-level environment with such kind people, and it's such a relaxed environment as well. Matt, for letting me Photoshop you and show it to 50 people. But also, I know it's not easy to deal with a 17-year-old bumbling around in a lab with all this expensive equipment around. But I really appreciate your patience and guidance. I really learned a lot from you. The education outreach team, Donna and Madison, and all the scholars as well, thank you for making the summer so fun. And last but not least, everyone who came out today, my parents, my friends, and a special shout out to Mrs. Lindsay Shaman for making a special effort today to come here. Thank you everybody for coming and listening to my presentation.
Excellent job.
Thanks.
Nice one.